All right, it's my pleasure to introduce Deil. He is a Fulbright Fellow and a PhD student in the Fisheries and Wildlife and Environmental Toxicology at Michigan State University. His research focuses on understanding the impact of PFAS contamination on overall health, particularly on disease susceptibility and survival of prominent salmonid species in the Great Lakes. Take it away.
Thank you, Nick, for that introduction. Hi, everyone. Before I start, I would like to thank my collaborators for this project. Without their help, it's not possible. For this afternoon, we will talk about the influence of perfluorooctane sulfonate on the host pathogen interactions and disease outcomes in juvenile lake trout. So let me give you a little background about PFAS. PFAS, which stands for per and polyfluoroalkyl substances, which is a mouthful, is a very large group of nearly 15,000 synthetic chemicals that have been first identified in 1940s. These compounds possess highly desirable properties. They make materials resistant to water, heat, oil, and they are extremely stable. And because of these characteristics, they have been widely used in modern applications and have become an integral part of industrial processes and consumer products worldwide. In fact, most of us come into contact with items containing PFAS every day. Chances are you have something beside you right now that contains PFAS, such as your clothing, your food packaging, waterproof materials, or even your electronics. So unfortunately, these characteristics that made them desirable are also the very reasons why they are become a problem in the environment. Since they are extremely stable and difficult to break, they stay in the environment for a significant long period of time. That is why they also often called as forever chemicals. And the most alarming of all, most of these compounds ends up in freshwater bodies, affecting the associated flora and fauna. So since 1940s, thousands, if not millions of products have been manufactured using PFAS in their production. You can also see PFAS in your cooking pans and shoes, among others. And in early 2000s, many people finally noticed that these chemicals are very toxic, which eventually led to the phase out of legacy PFAS, which is PFOA and PFOS. So mind you, it was phased out in 2000. However, companies just developed PFAS alternatives with the same properties, which eventually led to similar environmental problems. So one thing that all these compounds have in common is that they contain a chain of carbon atoms bonded to fluorine. Now, the strongest single bond in chemistry is actually the carbon-fluorine bond. This bond is one of the reasons why PFAS are very difficult to break. And there is no natural process in the environment that could degrade PFAS. Therefore, once present, they accumulate in soil, water, and even in plants and animals. So this is just like in 2022, a study have identified the potential contamination sites here in the US, and major sites were military sites, airports, wastewater treatment plants, and industrial facilities. These sites are a major source of PFAS because they use firefighting foams, which contain a plethora of PFAS compounds. And since most PFAS end up in freshwater bodies, it's no surprise that we are frequently detecting it in the Great Lakes. In the Great Lakes, PFAS is found accumulating in various species. The graph on the left shows the concentration of the 10 different PFAS in each of the Great Lakes. And the graph on the right shows the concentration of total PFAS within the Lake Michigan fish. You can see that even though PFOS and PFOA was phased out in 2000, we are still detecting these PFAS compounds up to now. It means that they are really stable and they don't break down easily. Second, notice that the concentration of PFAS in the fish is much higher than the concentration that we see in the water, which highlights the bioaccumulation potential of PFAS to reach higher concentrations within tissues in associated biota. So given the high concentration of PFOS in this case, detected in fish population across the Great Lakes, evaluating the PFAS on fish, particularly in fish health, is very essential. So the objective of this study is to evaluate the influence of PFOS, the most dominant PFAS, on overall health and disease susceptibility of lake trout. Specifically, we want to determine if PFOS exposure can increase the disease susceptibility and disease progression and mortality of lake trout challenged with EEDV. Lake trout was selected because it is a valuable indigenous keystone predatory fish in the Great Lakes, while EEDV was chosen as the pathogen of focus as it poses a major threat to lake trout rehabilitation programs in the region. So first, we condition our fish. We use juvenile lake trout. Average weight is 122 grams, and the age is 2 years old. We use flow-through polypropylene tanks. We condition them for 2 weeks in these tanks prior to the start of the experiment, and we kept them in 10 degrees Celsius water, where they also fed ad libitum. So after 2 weeks, we did the PFOS exposure. So during the PFOS exposure, there are like two groups, the controls which are not exposed to PFOS and the groups that are exposed to PFOS. The concentration of PFOS that we use is 1 milligram per liter. Since this concentration was reported to disrupt immune-regulated genes, and exposure happened for 96 hours using a flow-through system, which has a flow rate of 100 to 100 ml per minute. And we also fed them ad libitum during this period. So the problem in this kind of experiments is like we're producing so much PFAS waste every day. In this experiment, in a day only, we produce 900 liters of PFOS, and there's no commercial filters that could filter that out. And we are not allowed to dump that PFOS water in the sewer. The state will sue us. So we develop our own filters in our lab, and it was tested that after the filtration, PFOS and other PFAS are undetectable in the wastewater. So after the 4 days PFOS exposure, we applied the treatments and we did the disease challenge. There are four treatments in this study: control and PFOS. These two groups are not exposed to disease, and the other one is EEDV. It was only exposed to the EEDV or the disease. And the last one is PFOS and EEDV, which is exposed to both PFOS and EEDV. The concentration of the virus we use is 4.7 times 10 raised to the 4th power. This is actually the LD50 for lake trout. And we maintained the fish in this concentration of the virus for 1 hour at 9 degrees Celsius, since this method has been shown to induce clinical morbidity and mortality in juvenile lake trout. So after that 1 hour disease challenge, we returned the fish to these polypropylene tanks. We observed them for 2 months. We maintained water temperature at 9 degrees Celsius, and we collected samples regularly for the monitoring of disease progression. And we analyzed the samples using qPCR. Now, the problem in starting the study is like we don't know if the fish will survive the PFOS exposure, and we don't know when to do the sampling. That's why we did the preliminary study. So in this preliminary study, it provides us information that the fish can actually survive the 4 days PFOS exposure of 1 milligram per liter, and it also provides us specific sampling points where to do the sampling in the main study. But here you can see the trend of the viral load in the preliminary study. Here you can see a delay in the viral load in those exposed to PFOS and disease, but after 21 days, there's a pronounced increase in the viral load in those exposed to PFOS and disease, while in those group exposed to EEDV only have a steady increase of viral load. In the main study, there's a steady increase of EED copy per milligram of tissue between day 14 and day 21 in treatment groups challenged with the virus. The highest viral load was recorded in day 21, similar to what we have observed in the preliminary study, after which the viral load gradually decreased until the end of the experiment. When comparing fish exposed to PFOS with those that were not, the PFOS-exposed group consistently exhibited higher viral loads throughout the study period. Additionally, the decline in viral load after day 21 was less pronounced in the group exposed to both PFOS and the disease as compared to the group exposed to the disease alone. Meanwhile, EEDV copies were detected in the control and PFOS group, as you can see here. This detection of EEDV DNA in healthy fish was also reported in other studies, but this could actually attributed to a lot of factors, including the possibility that the control fish were already carrying low levels of the virus prior to the start of the experiment, or even cross-contamination during daily fish maintenance. We also analyzed skin samples. We found a similar trend in viral load when processing the skin samples. The highest viral load was also recorded in 21 days post-infection, consistent with our also preliminary study. The peak viral load also occurred. We also observed a much higher viral load here than those found in fin samples. By day 28, the viral load decreased, followed a slight increase between days 42 and 56. When comparing the group exposed to both PFOS and EEDV with the group exposed to EEDV alone, PFOS-exposed fish consistently exhibited higher viral loads, except on day 56. For the survival, no mortalities were observed in the control and PFOS groups. Mortalities were only observed in fish challenged with the disease, with most occurring between 30 and 49 days post-infection. And after the experiment, the average survival rate for the EEDV exposed group was approximately 66%, while the group exposed to both PFOS and disease have a survival rate of only 33%. We also checked if the surviving fish were also infected and whether the deaths during the study were caused by EEDV. To confirm this, we used qPCR to test fin samples for both live and dead fish for the presence of the virus, and analysis showed that the surviving fish in the control and PFOS group are negative to EEDV. All samples from the EEDV group showed 91.67% infection rate, while the group exposed to both PFOS and EEDV had a slightly higher infection rate of 93%. So these are the observed clinical symptoms during the 2-month study. All these fish are from the group exposed to both PFOS and EEDV. So we observed fin erosion, erosion of caudal peduncle, multiple skin lesion, exophthalmia, and ocular reddening. When comparing the severity of clinical symptoms between groups exposed to both PFOS and EEDV and group exposed to EEDV alone, fish in those exposed to both PFOS and EEDV exhibited more severe signs, such as deeper skin lesions, more extensive fin erosion, and overall greater tissue damage. So in summary, our research shows that PFOS exposure increases lake trout susceptibility to EEDV. Fish exposed to PFOS had higher viral loads, more severe clinical symptoms, and greater mortality than unexposed fish. This result suggests that PFOS weakens fish immune system and making them more vulnerable to viral infection. Given PFOS persistence in aquatic environments, this highlights an important ecological management concern. Pollutant exposure may amplify the impact of existing fish diseases. And I would like to acknowledge the GLFT for funding this research, and also the Fulbright Philippines for allowing me to pursue my studies here in the United States. Thank you.
Interesting and important work, Deil. Thank you for sharing. Folks, if you have any questions, feel free to unmute yourself, or you can drop them directly into the chat. Okay, we got a couple coming into the chat. The first one, I'm curious about how you could mitigate the concentration of PFAS in the water.
When you say mitigate, like control or remove during the experiment? Is it like a general? So in the experiment, we developed a dosing system. So if we will calculate the amount of PFOS that we need like 900 liters of PFOS in one day. So what we did is like we mix a very concentrated one. It's like 337 milligrams per milligram per liter. That's the concentrated one. And inside, we mix it using peristaltic pump with fresh water. So there's like 4 pumps working around mixing PFAS and dosing the PFAS tanks.
Great. And then how did you determine the concentration of PFAS that
So
you used?
so we have here in MSU we have a PFAS analytical system. They haven't yet analyzed the waters, but I send them the samples for the PFAS concentration and even the PFAS stock concentration and the PFAS samples from the dosing tanks. So I also collected tissue samples and we're still just waiting for the results for the analysis.
Okay. And then Cheyenne's got another question here. In the ultra concentrated solution, did you have any problem solubilizing the PFAS?
So the problem with high concentration of making high concentration of PFAS is the formation. Now, one thing that we did is like after we developed or mixed the high concentrated PFAS, we put a pump inside that bucket and allow it to mix for 2 to 3 days. And that's before we use it for the main experiment. So it's very difficult to dissolve it in the water. Before we are using the MSO, but now that's not allowed anymore. But not so far that's the only thing that we are doing. And we're still waiting for the PFAS analysis. So hopefully we reach what we aim. And one thing too is based on our experience, if you are preparing 1 milligram per liter concentration, when they analyze it, it's only like half of that. That's why when we make PFAS, we are accounting for that loss. So in this case, we are making it more concentrated to make sure that we reach that concentration that we want.
Great. And then one last question. We'll take it from Tom Jones. Did you look for EEDV in any of the internal organs?
Yeah, actually, I collected liver samples. So EDV it attacks the integument of the fish. So we are very curious if those exposed to PFOS have like internal infection. So we collected liver, and we are still in the process of processing that one. But we collected liver samples. So hopefully in the next coming weeks we will have the results for that.
That sounds good. And hopefully you're back next year to present the update.
Thank
Thanks
you.
again, Deil.